project for the machine learning course at Georgetown, and I'm talking about brain tumor subtype exploration for candidate biomarkers. And I chose this project because I'm interested in the use of machine learning effectiveness clinically. In this original research project specifically, the authors achieved a 90.1% accuracy with a random forest model of categorizing 5 different brain cancer subtypes. And my goal is to match or exceed that accuracy and evaluate how the model can distinguish between different types of brain cancer, and also if we can use that to distinguish between cancer versus the same model to distinguish between cancer versus normal tumor types. And so that is a challenge that I looked at solving, is classifying those 5 pediatric brain cancer subtypes from gene expression levels that were captured from the original authors of this study, and then classify those cancer and non-cancer samples. And then if we can successfully distinguish between the different cancer subtypes, identify the significant genes so that we can expand future work to use those genes as biomarkers for immunotherapy. And so the hypothesis is that the accuracy will be able to be matched. There's already an existing random forest model of this data with a 90% accuracy rate, so the goal is to meet that. However, I'm not 100% sure if we can distinguish between cancer versus non-cancer, or just between the different types of brain cancer that we're looking at. So we'll see what happens with the broader application there. But then I do believe that with distinguishing between the different subtypes of brain cancer, we'll be able to identify the common genes among the cancers that can be used as candidate biomarkers moving forward. And so a little bit of background on this. Brain tumors were the leading cause of death for childhood cancer in 2013, and that was the impetus for the original author's work, where they collected different brain tumor samples from different children and recognized that they have different immunophenotypes. And so the goal of the original study was to try to treat those different brain cancers by targeting the immunophenotypes. And the data was aggregated and a random forest model was applied to it that found a 91% accuracy. And that came from a different source. So the goal here is to reproduce those results that were originally found in that. And so what I did was I first explored the data using PCA and heat maps to try to get an overall big picture of what the data looked like. And with the PCA, I had found that there wasn't any clear distinguishable groupings with the different brain samples or within the different non-tumor normal samples. So it didn't look very promising initially. So I used lazy predict to try to run a large number of classification models all at once and get an estimate of which would be able to work with the data most successfully. And lazy predict actually predicted random forest to be one of the top 5 models. So I ended up proceeding with that classification method. And from there, I looked at which features were deemed significant in the random forest classification. And here's an example of the confusion matrix that came from using the random forest data. And we ultimately got a 97.4% accuracy. We can see the only misclassified cancer subtype. There's the one pilocytic astrocytoma. However, that also had the second fewest number of test samples after using a test train split of 30% of the test data. And so it's not too surprising that the results are off, but it's very beneficial to see that the results were so successful throughout for each brain cancer type, and for the normal non-tumor samples. And so the other part of the project that I did was I looked at all the combined data, so the 5 different cancer subtypes versus normal. I grouped all of the cancer subtypes into one cancer type and compared those to normal, and then did each individual cancer versus the non-tumor normal samples, to the glioblastomas, the medulloblastomas, the pilocytic astrocytomas, and the ependymomas versus normal samples, and looked at each feature that was deemed significant in the random forest classification and mapped those back to which genes they were representative based on the gene expression levels that were used. and then looked into the functions of those genes or the proteins that they coded for to see if there was anything in common. And it kind of appeared that there was no overlap between any of the individual brain cancers or cancer as a whole. So that kind of is indicative that these different brain cancer subtypes do have the different immunophenotypes, as was confirmed in the original literature that looked into this data, seeking to find the candidate biomarkers. So this investigation kind of validated those results and shows that we can potentially target these genes moving forward for immunotherapies. And that is the big takeaway, is that there is potential for pediatric brain cancer subtype biomarkers looking at these genes that were deemed significant in distinguishing each different brain cancer subtype from one another. And we were able to do that with the random forest model that had a 97.4% accuracy, and that accuracy was also applied when distinguishing cancer versus normal in the broader categorization. And so moving forward, future work can be done exploring these genes and the proteins that they code for the potential for biomarker targets for immunotherapy and the viability of those tools or of those genes to be used in tooling moving forward for treatment options. Thank you.